Welcome, colleagues, esteemed researchers and clinicians. On behalf of Mega Psi and the Gemini family to present Canine Geroscience and translational longevity models. Today, we bridge the gap between rodent laboratory findings and human clinical application. Welcome to the future of biogerontology. Welcome to the lecture!
We spend billions of dollars building these incredibly sterile, high-tech laboratories to decode human biological aging. You know, we sequence genomes, we isolate variables, we engineer these highly specific environments to try and understand how and why our bodies degrade. But looking at the translational data for this deep dive, the more you look at it, the more it seems the key to understanding cellular senescence and neurodegeneration might not actually be in some billion-dollar vivarium.
No, oh, go ahead.
I was just going to say, it might literally be sleeping at the foot of your bed.
Right, exactly.
Yeah.
It is a profound shift in how we approach geroscience, treating the domestic companion dog as the apex translational model for human aging. It forces us to reevaluate decades of standard laboratory protocols.
Yeah.
Because if you look at the comprehensive systematic evaluations of canine models and the actuarial data and the molecular pathway maps, you quickly realize that we've been missing a massive piece of the puzzle by isolating our models from the real world.
And for anyone listening who, works in a clinical setting or runs clinical trials or is just deeply invested in the mechanisms of health span, the way we currently model aging, presents a huge bottleneck.
Oh, absolutely.
Like, we've relied so heavily on genetically heterogeneous mice, like the UMHET3 strain, to map these age-related biological failures. But looking at the literature, those models seem to be hitting a wall when it comes to translating into human clinical success. Aren't those mice specifically bred for this? Like, why are they a bottleneck?
Well, the limitation stems from the biological vacuum we create for them.
Okay.
These mice are kept in artificial pathogen-free environments. They eat standardized diets. They experience zero environmental variance.
Right.
And crucially, they lack the spontaneous multimorbidities that define actual human senescence. I mean, if you want to study an age-related disease in a mouse model, you almost always have to chemically or genetically induce it.
Wow. So it's not even naturally occurring.
Exactly. You're studying a simulated disease in an organism with an immune system that essentially resembles a neonate's.
Oh, geez.
Because it has never fought off a natural pathogen.
Which fundamentally alters how their immune system ages compared to a human living in a dirty, complex environment.
Right.
And beyond the biological limitations, there's this staggering logistical barrier. If we want to run highly powered long-term aging cohorts, we run into the reality of late life attrition costs.
The economics are completely prohibitive.
Yeah.
Because rodent mortality increases exponentially in late life. The cost of generating a surviving healthy late life mouse rises non-linearly.
Because they just keep dropping off.
Yes. To maintain a single UMHET3 mouse to 32 months of age, assuming it somehow remains tumor-free.
Which is rare.
Very rare. The cumulative institutional husbandry cost is roughly $1,400.
Wait, $1,400 for one mouse?
For one mouse. So if you need a statistically significant cohort of thousands of these animals to test a subtle longevity intervention, you're burning millions of dollars just to keep the baseline subjects alive long enough to study them.
Okay, let's unpack this and look at the mathematical alternative, because this is where the actuarial calculus of the canine model becomes so compelling. The literature references the Gompertz hazard function, which models how mortality risk exponentially increases as an organism ages.
Right.
Now, dogs age about 7 to 10 times faster than humans. So we get this compressed 3 to 5 year longitudinal endpoint that maps directly onto human senescence. But the specific variables within that Gompertz equation paint a really counterintuitive picture.
They do. The paradox is in the slope.
Yeah, explain that.
If we look at the demographic aging rate, the Gompertz slope itself, which represents how quickly the mortality hazard compounds over time, it is technically lower in dogs than in humans.
Really?
Yeah, for female humans, that slope is about 0.089. For female dogs, it's significantly flatter at 0.0214.
So if their demographic aging rate is mathematically lower, we have to ask why their lifespans are so severely compressed. And it turns out their overall shortened lifespan is driven by a substantially higher initial baseline mortality hazard.
Exactly.
It's the biological cost of their rapid early development and the massive physiological variations we've bred into them. I mean, we're working with a species that demonstrates a threefold variation in longevity based entirely on intraspecific life history traits, primarily body size.
And that compressed timeline happens within our exact exposome.
Oh, that's right.
Unlike laboratory mice companion, dogs drink the same municipal water we do.
Right.
They are exposed to the same household volatile organic compounds, the same circadian disruptions from artificial light, the same microplastics in the environment.
They're literally sharing our world.
Sharing our exact world.
Yeah.
Which is why the emergence of citizen science biobanks is transforming the field. Initiatives like the Dog Aging Project, which is tracking over 50,000 community dogs.
Wow.
And the Mars Petcare Biobank, tracking 20,000 pets. They're finally allowing us to capture real-world environmental data alongside deep molecular profiling.
And there's a specific data point from that Mars Petcare Biobank that any clinicians listening to this should really pause and consider.
Oh, the baseline tests.
Yeah. When researchers analyzed baseline blood tests from dogs that primary care veterinarians had deemed entirely healthy based on physical exams, up to 17% of those results fell outside established reference intervals.
It illustrates a critical vulnerability in how we practice medicine across both species.
Totally.
Because relying on rigid static reference ranges means we are consistently missing the subclinical transition into disease.
Right.
If a patient's liver enzymes double over 2 years but remain just under the upper limit of normal, a static reference range calls that healthy.
But it's obviously not.
No.
Yeah.
A dynamic individualized biomarker framework recognizes that as a massive biological alarm bell. We have to stop waiting for an organism to fall off a metabolic cliff before we intervene.
Right. And if we're sharing this incredibly complex environmental exposome and it's driving subclinical metabolic changes, we have to look at how that environment impacts the most vulnerable biological network of all, right, the central nervous system, which means examining how the canine brain degrades.
Yes. Aging companion dogs spontaneously develop a progressive neurodegenerative syndrome known as canine cognitive dysfunction syndrome, or CCDS.
Right.
And what makes this so vital for researchers is the histopathology. CCDS is essentially the early beta-amyloid predominant counterpart to human Alzheimer's disease. It provides a natural window into the mechanics of cognitive decline.
So let me make sure I'm visualizing the mechanics here properly, especially regarding clearance.
Okay.
In a healthy state, the brain relies on the glymphatic system.
Yes.
We have these specialized water channels called aquaporin 4 or AQP4, and they sit on the astroglial end feet that wrap around the brain's microvasculature. So are these channels essentially acting like an overnight fluid exchange network? Like a city's street sweepers, utilizing the convective flow of cerebrospinal fluid to wash away soluble beta amyloid and metabolic waste while they sleep?
That is an excellent way to conceptualize it.
Yeah.
And the mechanical breakdown of those street sweepers is where the pathology begins.
Oh, okay.
So with advancing age, the vascular system undergoes progressive arterial stiffening. The elasticity of the vessels diminishes.
Right.
And that mechanical stiffness at the vascular level directly causes a loss of the AQP4 polarization you just described.
Yeah.
The astroglial end feet physically lose their structural integrity and their ability to pump that fluid.
So the localized clearance network simply fails.
It fails, and it triggers a devastating positive feedback loop. Without that convective clearance, you get a paravascular accumulation of unstructured beta-amyloid oligomers.
Just building up like trash in the streets?
Exactly. As these proteins build up around the vessels, they drive a condition called cerebral amyloid angiopathy, or CAA. The amyloid physically invades the blood vessel walls, which disrupts the blood-brain barrier, restricts oxygen delivery to the surrounding tissue, and ultimately leads to microhemorrhages and widespread white matter myelin loss.
And we can trace that specific neurophysiological decay to very distinct clinical behaviors, right? Veterinary medicine uses the DISHA framework to diagnose this, and it maps beautifully to the localized brain atrophy.
Yes, it does.
For example, the D stands for disorientation, so dogs getting trapped behind furniture or just staring into corners.
Yeah.
Structurally, that correlates with severe atrophy of the prefrontal and parietal cortices.
Right. And we see similar direct correlations with the S, which is sleep disturbances.
Oh, right.
Nighttime pacing and vocalization aren't just behavioral quirks. They represent the physical degeneration of the suprachiasmatic nucleus.
Wow.
Which completely destroys their circadian rhythmicity.
And then the H for house soiling and memory loss maps directly to bilateral hippocampal atrophy and a severe drop in synaptic density. Now, looking closely at the histopathology in the literature, there is a fascinating distinction. Dogs generally do not form the highly structured, dense core fibrillar plaques, nor do they develop the extensive intracellular neurofibrillary tangles that define late-stage human Alzheimer's.
Right.
Their amyloid burden remains mostly diffuse. So I have to ask, since they don't form those late-stage markers, does the absence of those actually make the canine model more valuable for testing early interventions?
It is arguably their greatest asset as a model.
Really?
Yes. By lacking those end-stage fibrillar plaques and massive tau tangles, CCDS perfectly isolates the early amyloid-driven neuroinflammatory phase of the disease.
Oh, I see.
In human trials, we frequently fail because we intervene when the neuronal death is already massive and irreversible.
The damage is already done.
Exactly. The canine model gives researchers a critical, naturally occurring window to test disease-modifying therapeutics during the exact interventional period human medicine has been desperately trying to target.
That makes so much sense. But the brain's failure to clear amyloid isn't happening in isolation. If the local astroglial networks are failing, we have to look at what's happening to the body's systemic defense network at that exact same time. The neurodegeneration is intimately linked to the collapse of the immune system.
Right. And to understand that systemic collapse, we have to look inside the bone marrow niche.
Okay.
The foundation of immunosenescence begins with hematopoietic stem and progenitor cells, or HSPCs.
Right.
As these specific stem cells age, they undergo profound epigenetic and metabolic shifts. They acquire what we call a senescence-associated secretory phenotype, or a SASP.
So they essentially become toxic factories leaking pro-inflammatory cytokines into their own microenvironment.
Yes, and that localized chronic inflammation forces a biological pivot known as myeloid lineage skewing.
Myeloid lineage skewing, okay.
Right. To counteract the systemic inflammation, the immune system aggressively upregulates its production of myeloid cells, so macrophages, neutrophils.
Right, the first responders.
Exactly.
Right.
But stem cells have a finite capacity.
This massive myeloid expansion comes at the direct expense of lymphoid progenitors. So the production of naive T cells and B cells plummets, leaving the adaptive immune system fundamentally compromised and unable to respond to novel antigens or mutated cells.
And to study this phenomenon without the background noise of different commercial diets or varying exercise routines, researchers utilized the Vika Sled Dog Project.
That's a great study.
Yeah, they analyzed a cohort of retired Alaskan sled dogs living in highly uniform conditions. And the data revealed massive age-dependent accumulations of a very specific problematic cell type, right? Polymorphonuclear myeloid-derived suppressor cells, or PMN MDSCs.
Yes. These PMN MDSCs are highly immunosuppressive. They actively inhibit T cell activation and proliferation.
Oh, wow.
As they accumulate in the peripheral blood of older dogs, they suppress the very mechanisms meant to identify and destroy abnormal cells.
Right.
They create a systemic pro-tumorigenic microenvironment. This is the biological reality of inflammaging, which explains a glaring statistical reality in veterinary oncology. And here's where it gets really interesting. Nearly 50% of dogs over the age of 10 die from cancer.
It's staggering.
It is. And these are not artificial lab-induced tumors. They are spontaneous malignancies that share remarkable genetic homology with human cancers.
Absolutely.
For example, canine osteosarcoma follows the exact same aggressive clinical course driven by the same TP53 tumor suppressor mutations as pediatric osteosarcoma in humans.
Right.
And canine mast cell tumors are frequently driven by the exact same KIT receptor tyrosine kinase mutations that we see in human gastrointestinal stromal tumors.
It really solidifies comparative oncology as a highly predictive model.
Yeah.
But the neoplastic risk is just one consequence of a much broader biological collapse. As these organisms approach mortality, they share a very distinct metabolic failure network with aging humans.
Right.
The decline doesn't happen simultaneously.
Yeah.
It is a cascade where the failure of one system poisons the next.
Right. So if we trace that cascade, it frequently begins with renal decline.
Yes.
As the glomerular filtration rate drops, the kidneys fail to clear metabolic waste. You get a systemic accumulation of uremic toxins like guanidino compounds and creatinine circulating in the blood.
And those circulating toxins create intense cellular stress, which directly impairs lysosomal function.
Okay.
The cells lose their ability to clear damaged proteins, leading to a profound proteostasis collapse.
Right.
When we look at the plasma metabolome, we see a massive surge in post-translationally modified amino acids, or PTMAs. Molecules like N-terminally acetylated phenylalanine build up exponentially because the cellular degradation machinery is just overwhelmed.
And as that cellular garbage accumulates, it directly impacts bioenergetics. The mitochondrial networks begin to fail. We see a significant accumulation of long-chain acylcarnitines in the plasma. To use an analogy, imagine a factory where the loading dock is completely overflowing with raw materials, in this case fatty acids, but the internal assembly line has broken down.
That's a good way to picture it.
Yeah, it's like an engine flooding because the mitochondria completely fail to process the lipid fuel, forcing the entire cell into a state of metabolic inflexibility.
Which ultimately culminates in severe bioenergetic depletion. The system experiences a systemic drop in nicotinamide, the primary substrate for the NAD+ salvage pathway.
Oh, right, NAD+.
Yeah, and without adequate NADP+, the sirtuin proteins, which are the master regulators of DNA repair, inflammation, and cellular survival, they are fundamentally disabled.
Wow.
When you see the specific metabolic signature, renal toxic proteostasis, collapse, mitochondrial stalling, and NAD+ depletion, you are looking at a highly accurate predictor of short-term mortality in both dogs and humans.
So with that cascade of biological failures so clearly mapped, the critical question becomes, how do we intervene?
Right.
How does geroscience actually disrupt this failure network? We can see the cutting edge of this by looking at how veterinary medicine is addressing cardioprotection and sarcopenia.
Yes, the clinical progression of myxomatous mitral valve disease, or MMVD, offers a perfect illustration of this. It is a naturally occurring cardiovascular disease affecting up to 80% of geriatric small breed dogs.
80%, wow.
Yeah. As the mitral valve structurally degrades, the heart is forced into chronic volume overload. By the time the patient reaches ACV stage C, which is decompensated heart failure, the metabolic demands on the myocardium are completely unsustainable.
Right, because of that mitochondrial stalling we just discussed, the failing heart can no longer process normal lipids.
Exactly.
It's forced into metabolic reprogramming. It switches its primary fuel source to ketone bodies, specifically beta-hydroxybutyrate, just to maintain contractility.
And while the heart is desperately scavenging for alternative energy, we see a concurrent spike in 3-methylhistidine.
Okay, what does that indicate?
This is a vital biomarker because it indicates active myofibrillar protein degradation.
Oh, no.
The failing heart is essentially cannibalizing the body's skeletal muscle to survive. It is driving cardiac cachexia, which directly links the localized cardiovascular failure to systemic sarcopenia, the severe age-associated wasting of muscle tissue.
And that wasting is exacerbated by a massive drop in anabolic hormones like IGF-1, combined with the systemic inflammaging from those myeloid suppressor cells we talked about earlier.
Right.
The inflammation literally activates the ubiquitin proteasome pathway to break down muscle fibers. But veterinary researchers are no longer just documenting this decline. They are actively testing therapeutics to reverse it, leveraging a highly innovative regulatory mechanism called the FDA's Expanded Conditional Approval Pathway, or XCA.
The XCA pathway is arguably laying the regulatory blueprint for future human preventative geroscience.
Yeah, it's fascinating.
It allows therapeutics targeting complex unmet medical needs to receive conditional market authorization once they demonstrate a reasonable expectation of effectiveness and meet rigorous safety profiles.
Right.
So it accelerates the deployment of disease-modifying drugs into the clinical population while long-term efficacy data is still being gathered.
And the therapeutics entering this pipeline are incredibly sophisticated. Looking at the small molecule interventions, we have the TRIAID trial. They're utilizing weight-adjusted low-dose rapamycin in middle-aged dogs. The goal is to selectively inhibit the mTORC1 pathway.
Right.
By doing so, they aim to mimic the profound cellular benefits of caloric restriction, specifically triggering autophagy to clear out that cellular garbage we mentioned, without inducing the severe immunosuppressive effects associated with higher organ transplant doses.
We also see targeted metabolic modulators like LOY-002, designed to reverse age-associated insulin resistance in senior dogs.
Okay.
By restoring insulin sensitivity, the intervention mitigates a primary metabolic driver that accelerates both neoplastic growth and cognitive decline.
Right.
And we must highlight the paradigm-shifting nature of LOY-001 and LOY-003, which directly target the IGF-1 axis disparity.
Yeah, because large breed dogs overexpress IGF-1 during development, which drastically accelerates their cellular aging, causing them to live nearly half as long as small breeds.
Right.
LOY001 targets that exact mechanism, and the FDA's decision to grant it a reasonable expectation of effectiveness represents the first time a regulatory body has formally recognized lifespan extension as a valid, treatable clinical endpoint.
It's huge. But if we want to see the absolute frontier of the Apex translational model, we have to look at gene therapy.
Oh yeah, RJB01.
Exactly, RJB01. This is a dual gene therapy delivered via an adeno-associated virus, or AAV vector, specifically engineered to treat the MMVD heart failure we discussed earlier.
The biological mechanism of RJB01 is fascinating.
It really is.
The AAV vector delivers two synergistic genes directly into the target tissue. First, it upregulates the expression of FGF21, a hormone that profoundly enhances systemic metabolic function, improves insulin sensitivity, and reduces chronic vascular inflammation.
Right.
But the second target is what really alters the disease pathology. It expresses STGF-beta-R2.
Yes, and that is a soluble decoy receptor.
Decoy receptor, right.
In a failing myocardium, TGF-beta1 is the primary cytokine driving tissue fibrosis. It activates local cardiac fibroblasts, compelling them to lay down excessive extracellular matrix, which turns healthy, pliable heart muscle into rigid, non-functional scar tissue.
Wow.
By introducing a soluble decoy receptor, the gene therapy effectively creates a molecular sponge. It binds and neutralizes the circulating TGF-beta1 before it can attach to the fibroblasts, directly halting the fibrotic cascade at the cellular level.
And the clinical data from the trials with Cavalier King Charles Spaniels is remarkable. A single intravenous injection demonstrated durable transgene expression for nearly 3 years.
3 years, yeah.
When they combined this gene therapy with the standard of care drug pimobendan, it delayed the progression of heart failure by over 800 days compared to the placebo group.
800 days is incredible.
It is. The echocardiograms didn't just show stabilization, they demonstrated active structural regression, like the size of the enlarged left atrium actually reduced. The myocardium was essentially remodeling itself back toward a healthy state.
Synthesizing all of these data points from the neurophysiology of the glymphatic system to the metabolic collapse and these advanced gene therapies, the overarching thesis becomes undeniable.
Yeah.
Companion dogs do not merely simulate isolated variables of human aging in a sterile vacuum. They experience the exact same multimorbidities driven by the exact same metabolic failures within our shared environment on a clinically actionable timeline.
They provide a real-world, highly predictive model for preventative geroscience. Whether it's utilizing citizen science biobanks to prove the necessity of dynamic biomarkers or leveraging FDA pathways to test mTOR inhibitors and AAV vectors in naturally aging populations.
Ultimately, this research forces a philosophical shift in medicine. We're moving away from treating aging as an inevitable collection of separate end-stage diseases.
Right.
We're learning to treat aging as a fundamental, modifiable biological mechanism. The blueprint for extending human health span is currently walking on a leash beside us.
It really is. And I want to leave you with a final, slightly provocative thought to consider regarding that blueprint. We discussed how centuries of selective breeding created a threefold variation in canine lifespans almost entirely linked to traits like body size and the IGF-1 axis. We artificially bred large dogs to age faster, and now we're using advanced small molecules and AAV gene therapies to try and correct those biological extremes.
Right.
If we're currently proving that we can manipulate these core longevity axes in a complex mammal, how long until human longevity researchers look at our own genetic variations and propose using CRISPR to artificially engineer a biological small dog phenotype directly into the human genome, rewriting our baseline mortality hazard from the moment of birth?
Thank you for being with us. Mega Sci is for you and for us.